afternoon. Welcome to another edition of the DeFord Lecture Series. We're very, very happy today to have Dr. Jessica Guo from currently University of Arizona, and to tell us a little bit more about Jessica's work and what she's going to be talking to us about today is Ashley Matheny.
Hi all, thank you so much for making time for us this afternoon. I am honored to present for you my colleague, Dr. Jessica Guo. Jessica received her PhD in Biological Sciences from Northern Arizona University in 2019. Afterwards, she moved on to Utah, where she worked in Gabe Bowen's lab group studying plant isotope interactions. And now she works as an ecophysiologist and data scientist at the University of Arizona. And she has studied and continues to study plants and dryland ecosystems and the deadly combination of heat and droughts. So Jessica, take it away.
Okay, mic check. Can you guys hear me? Alrighty. Thank you for that warm welcome, Ashley. Thank you for having me. I'm very excited to be here and to talk to you guys about plant water use strategies in a changing environment. And so right off the bat, I'd like to acknowledge my co-authors in this work, a whole list of them, including Ashley, as well as other collaborators, which include field technicians and technical expertise and modeling help. And also acknowledge the land on which my research took place, including the Desert Botanic Garden, the Wasatch Range, and Bears Ears National Monument. And so fully cognizant that today I'm speaking to a geoscience department, I want to start off with a very broad cartoon of the water cycle and situate my place in where my research fits in the water cycle. I study one of the two main biological components of the water cycle, one being us as humans, and the other being this little arrow here, plant uptake or plant water use. So the whole rest of the water cycle I leave to you guys, but I am going to speak more about plants and the water cycle today. So, at the risk of stating the obvious, plants are non-motile, meaning they don't move. We all knew that. One of the reasons that I start here is because it's one of the first things we learn in biology about cells, animal cells versus plant cells. When you look under the microscope, maybe the first thing that jumps out at you is, okay, chloroplasts, right? Sites of photosynthesis. We probably, learned that equation pretty early on. And then the second thing are these cell wall structures. So plant cells are living cells situated inside these walls made of cellulose and lignin. And in a healthy leaf that's turgid, so that's upright and the opposite of wilted, the cell is actually putting positive pressure on that cell wall. It's pushing against the cell walls, and that's the healthy leaf that we know. If we remember the plants in our office, that should be what they look like. In other tissues, there are specialized cells whose job it is to grow and then die, and then leave behind pipe-like pieces of cell walls that are joined end to end for water to be able to be conducted through the plant in transpiration. So I show both of these because this is the reason why plants are non-motile, right? They have really rigid cell walls. It allows for structure, allows for trees to grow many meters, hundreds of meters into the air, but it prevents them from moving. They are rooted in place. But lest you think that plants are passively members of the water cycle, they are living. And so they are able to actively regulate how they respond to their environment. And one of the most, perhaps, easily identifiable from the outside ways that they do this is that some plants have sectoriality, so they can choose under different conditions to kill off parts of their branches and retain others in response to, say, a bad year, a bad drought. But that is only one of the tools in a plant's arsenal in order to respond to the environment. There's other things going on that we can't see with the naked eye going on inside those plants. And also at different time scales. And so on the left here, I'm showing a picture in SEM of stomata. Stomata are made up of two guard cells, and they are the pores on the leaf surfaces that allow for the exchange of carbon and water. So water is evaporating from the wet insides of the cells into the dry air, and they do that in order to so that CO2 can diffuse in and be the substrate for photosynthesis. And I want to talk about time scales specifically because stomata open and close on the order of seconds to minutes. So in the time it takes for a cloud to cover up the sun, or for the air to get really humid and heavy before a rainstorm, stomata can respond pretty instantaneously, let's say, to those external stimuli. But they're also integrating signals from within the plant. Earlier I talked about the cells that are living cells that are pushing against the cell walls for trigger. Plants are integrating that sensing and integrating that water stress. They're also incorporating feedback from plant hormones, which might be sending signals from other parts of the plant. But on the large scale, on a scale that, that a lot of us can immediately see, we think about kind of the climate change type drought causing large scale mortality events. This is spatially extensive, very obvious to scientists and non-scientists alike that this is a problem. This is a global change problem. And in fact, the 2002-2003 drought in the Four Corners area that killed off a lot of pinions but left the junipers healthy behind kind of spurred a lot of research in this plant hydraulics field. I'm particularly interested, or my research is situated at the time scales between these two, because the scene on the right, the pinions didn't die in one day. They didn't die because of a single weather or meteorological event. But what happened between the stomata opening and closing in response to something as like a cloud covering the sun, and that, which is a field of dead pinion, but the junipers are healthy. And I termed this the intermediate scale, where I was also borrowing a term from meteorology, the synoptic scale, so somewhere on the order of days to weeks to maybe months and seasons. What's going on? What is the cumulative stresses the plant experiences and deals with, and how does the plant biologically deal with those stressors to lead to the outcomes that we see on the right? Okay, so the main players, the main characters in my story today include stomatal conductance. Oh, let me back up. I'm going to talk a little bit about the soil-plant-air continuum. It's abbreviated SPAC. It's just kind of fun to say sometimes. And the stomatal conductance is the first character I'm going to introduce. It's abbreviated GS, although I tried to take out the abbreviations as much as possible. And that's referring to those stomata that I showed a picture of on the last slide, how open or closed they are. And stomata in these Western ecosystems where light is not very limiting, the main driver of how open or closed they are is something called vapor pressure deficit, or the dryness of the air. In case you're not familiar with this term, this is the difference between saturated vapor pressure and actual vapor pressure. So it's how much more water could that air at that temperature be holding. So it's a combination of temperature and relative humidity. But I also mentioned that stomata also integrates signals from within the plant, including plant water status, which we measure as plant water potential, abbreviated by the Greek letter psi. And the plant water status is intimately connected with the amount of water in the soil, so soil water content. And there is also feedback between the two, because depending on how open or closed the stomata are, and that water evaporating from the wet insides of the plants, that water status is going to drop, right? It's going to get less wet inside the plant because there's more water evaporating out. Both of these parameters, stomatal conductance and plant water potential, vary on a quite rapid basis, so on the order of hours, minutes to hours to days. They have known diurnal cycles. And it can be hard then to disentangle kind of more immediate things. Oh, a cloud came over and covered up the sun. That's why that happened. From kind of the longer term synoptic scale phenomena that add up to something like dead pinions on a landscape. So where my research is really focused is on plant water use strategies. How do plants regulate how they deal with the environmental signals that they are rooted in place and have no way of escaping from? So plants have to, in order to continue surviving, they have to balance that carbon gain and water loss. They can't just close their stomata all the time because they wouldn't be able to take in enough CO2 to produce carbohydrates. And with extreme climate and water stress, that balancing act becomes much tougher. And my question is motivated largely by, are these plant strategies themselves sensitive to these environmental controls? Are plants always doing or regulating their interaction with the environment similarly across seasons, say? And to tell this story, I'm going to tell this story in 3 vignettes. The first is looking at stomatal sensitivity in riparian trees. And shown on the bottom here, that's a cottonwood in the fall turning yellow. And the second, I'm going to talk about water potential regulation in creosote bush. That's the on the upper right. It's a very drought-tolerant shrub. And lastly, I'm going to connect water potential regulation to carbon fluxes in juniper species. And that's my colleague Steve Cannonberg up there installing an instrument in a juniper. I previewed this talk with an earth science colleague, and she said earth scientists love maps, and so I quickly scrambled to make a map for you guys. And I included Texas in here because I want to talk about the North American monsoon. Later on in the talk, I'm going to refer to monsoon and pre-monsoon, which it doesn't seem like, I don't think Austin experiences a North American monsoon. But this map here is showing in the colors the percent of annual precipitation that falls in July, August, and September, what we consider the monsoon season. And so the studies I just referred to happen to fall on this gradient. The riparian trees is up in the Wasatch Front. That the precipitation there mostly falls in the winter in the form of snow. My second study at the Desert Botanical Garden in Arizona. Don't be fooled by the fact that it's not as yellow as other parts of Arizona. The Sonoran Desert has bimodal precipitation, so a goodly portion of it falls in July, August, September, and there's also winter precipitation. And then Bears Ears, where my juniper work takes place in southeastern Utah there, is really on the very edge of the North American monsoon boundary. So some years there's a good monsoon and some years there's not. It's not very reliable. Okay. For my first talk, I'm going to focus on this beautiful picture on the left. This is a riparian ecosystem in what is otherwise drylands, in the drylands. And unsurprisingly, although they're not very spatially extensive, these riparian areas are thoughts of productivity, right? There's water, which means there's enough water for photosynthesis to happen. And there's also habitat. There's lots of species there and habitat for different kinds of other plants and animals. But I'm particularly interested in it because it allows me the opportunity to isolate the effects of soil moisture from the dryness of the atmosphere, or D. As you can imagine, most places, if the air is dry, the soil tends to be dry. If the air is wet, the soil tends to be wet during different times of year. But in this system, these riparian trees are drawing water from the water table, so the soil water isn't limiting, and I can use the dryness of the air to evaluate whether or not over time there's these kind of compounding effects of dry events. And this is particularly important because we don't necessarily have a good prediction of what the future holds for precipitation or groundwater in these regions. We do know that the air is going to get warmer, and warmer air can hold more moisture, so VPD, or vapor pressure deficit, is going to go up. And to do this, I employed an empirical model of stomatal sensitivity from Oren et al. All it says is that stomatal conductance, or GS, is equal to a linear function of log of vapor pressure deficit. And the intercept, that GS ref term, is a reference conductance, so how much stomatal conductance occurs at 1 kilopascal of vapor pressure deficit, and then M, a slope term. Stomatal sensitivity, or indexed as the M or negative M over GS ref. And in their survey and synthesis of a lot of different sap flux studies, they found that for mesic trees, most of the species, the S converged around 0.6, but for a couple of the species that were xeric shrubs, S converged around 0.4. So my question is really like, where do these riparian trees fall? Because they are mesic trees, but they're also living in a very dry place. And is there shifting in this S term across the season as the growing season progresses and the trees experience drier and drier air? So that was my question. How does sensitivity to D, or this S term, change over the growing season? And my first hypothesis, or instinctual gut feeling here, is that S should increase in all species, because as they experience these cumulative dry air events, the plants could lose conductivity, and then the plants would become more sensitive next time, right? Kind of like once or twice bursts. Oh gosh, what is that one called? I'll stop there. Anyway, they'll be more sensitive after that. Alternatively, seasonal changes in S could vary among species, reflecting different water use strategies. And to answer this question, I did a reanalysis of some existing published sap flux work. I could not do this without my colleagues Kevin Hulteen and Susan Bush. They went out in the summer of 2004 and instrumented 4 sites, 9 species, across an elevation gradient in the Wasatch Front. It takes a lot of work to keep those sites going, as I was just chatting with Ashley about before. They used the Granier or thermal dissipation method. And I wanted to look at both how these plants responded that same day to how dry the air was, as well as to the accumulation of dryness up through that day. So I had 2 metrics of atmospheric dryness. The first is the standard daily maximum D, and that's in the points on this figure. It's on the right-hand axis. And so at Jordan, for example, the lowest elevation site, unsurprisingly, it was also the driest, and some days the D reached about 6 to 7 kilopascals, which is pretty dry, very dry. And the second index or metric of atmospheric dryness I developed I called CDE, or cumulative excess vapor pressure deficit. This is an accumulation from day of year 1, how much the daily maximum D exceeded 1 kilopascal. And we're calling that 1 kilopascal as a reference conductance, or the level at which reference conductance is set. So it was designed to be kind of like growing degree days, if that's something you're more familiar with. And it's shown in the colored lines, and it's the monotonically increasing colored lines. And unsurprisingly, at Jordan, the lowest site, elevation site, CDE is both the highest and it has the steepest slope, right? It increases the quickest. There was a lot of dry days down in Jordan. The next thing I like to do when I'm testing these hypotheses and trying to design statistical models is look at the data. And so here I'm showing 4 different Russian olive trees on the axis of the Oren equation. So somatal conductance is on the y-axis, and on the x-axis is the log of D. And it is nice and linear, I think, but there's variation in there. It's kind of a cloud of points. And I went ahead and put my cumulative D excess or CDE metric in the color bar, and you can see that although the slope doesn't look that different between the purple and the green points, the intercept definitely shifted, and that the green is kind of got a lower intercept than the purple. So that means that later on in the season, after experiencing more dry events, these trees were able to have a higher conductance at 1 kilopascal. Well, that's a little bit interesting. That's not what I would have thought. But then there are other species, like box elder, plotted the same way, where it looked kind of the opposite. The green points early on in the season had a higher reference conductance, higher intercept than the purple points. So to examine this, I built a hierarchical Bayesian model for each individual and then the population, and I allowed the intercept term to vary. So I wanted the intercept term not to be one intercept, but that to vary with my CDE metric. And so that's just another, like a linear model within a linear model. It's another slope and intercept relationship between the two. And when I plot that, show that plot here, in the top at my lowest elevation sites, the relationship was either positive or not significant. So that means that at the lowest elevation site, the drier it got, or the more cumulative dryness there was, the reference conductance actually increased. Somehow they were able to increase their somatal conductance. While at the rest of the sites, the opposite was true. They were mostly 5 out of 6 species significantly decreased their reference conductance with more accumulated air dryness. And this GSREF, it was on the denominator of my sensitivity S, which I had my original question about. And because that GSREF varied in the season, my S also varied in the season. And just here again, a higher value of S is more sensitive. The somata close more easily, and lower values are less sensitive. The dotted line on these figures, they're all at 0.6 for each figure, although they're at different heights. That 0.6 is that what Oren et al. Found to be generally the sensitivity of mesic trees. And again, these are mesic trees, right? Because they look like mesic trees, they live near water, their roots are in water. But in fact, at the lowest elevation site, all of them behaved more like xeric trees, more like that 0.4 that Oren found for xeric shrubs. And for these two species, this is tamarisk and Russian olive, the S actually declined. That's not super obvious, but it did decline because of that positive value in the previous figure. The sensitivity here actually declined as the season progressed. They got less sensitive, which doesn't make a whole lot of sense. While the rest of the species either had maintained consistent sensitivity or had increased sensitivity. So my takeaway for this section is that seasonal changes in S are not uniform among species. And it emerged from the data that these two species that became less sensitive were actually non-native and ring-porous species. Ring-porous meaning that they have different variable vessel size diameters in a growing season. And that the other species became more sensitive to D. They were generally the native and diffuse porous species. Their vessel diameters remained consistent. And so the takeaway here really is that in a warmer future, where the D is going to be higher, in these desert riparian ecosystems, they're likely to decrease productivity because the plants are generally going to be more sensitive. They're going to spend more time with their stomata closed, and they can't photosynthesize under those conditions. And that might also lead to these changes in species assemblages that are going to be more dominated by these non-native taxa. Okay. Next, I'm going to move on to talking about water potential regulation in creosote. This was my dissertation work that I conducted at the Desert Botanical Garden. I want to start this section with an introduction to plant water potential, or SCI, in case you're not as familiar. And again, what's happening again here is the soil, plant, and air continuum. And so for water potential is how tightly the water is being pulled up through, with negative pressure, through the conducting tissues of the plant. And in order for water to move, it needs to go down the gradient, right? Just like chemical potential, other kinds of potential. So it's fun. When there is this gradient in this direction, water can go from the soil up through the plant and up into the air against gravity. Where we run into trouble is when the water potential of the soil drops. So that happens when the soil dries out. And if the plant didn't change its water potential, it would actually leak water back into the back into the soil. And that does happen. That's called hydraulic redistribution. But if that happened a lot, the plant would lose all its water and wouldn't photosynthesize at all. So the plant strategy generally is to also lower its water potential and to keep that water flowing up to the air for transpiration. But there is a consequence for this, right? In the tissues, an embolism can form because these water molecules are being pulled kind of one against each other. If you think about pulling on that water column really hard with these very negative water potentials, that column can snap, and that conduit will then fill with air. And that air breaks that continuum, and so no more water can be conducted through that particular pipe. If you build up enough of these, you can have runaway cavitation, and basically the hydraulic integrity of the tissue of this plant is just gone. And again, with no water being conducted, there's also not going to be any photosynthesis. So for the plant under water stress, this becomes a real balancing act. On the one hand, the plant could drop its water potential and continue taking up water from the soil, but that comes at the risk to its transfer capacity. It's risking those emboli that are going to block the conduits. On the other hand, a plant can maintain higher water potential, and that way there's not going to be as many emboli, and they can preserve its transport capacity. But if you do that, and the stomata have to stay closed, you're not taking up any water, and if you're not taking up any water, you're also not producing any sugars. So one way to think about these kind of choices that the plant has to make in deciding how to interact with the environment is this Anisohydrate or Isohydric Framework. So thinking about how the plant regulates water potential. And on one hand, we have Anisohydrate, where the water potential, so I talked about how they have known diurnal patterns. Water potential is generally highest right before the sun comes up, so pre-dawn, and lowest in the middle of the day. And in anisohydric plants, you have a pretty big difference between pre-dawn and midday because the stomata are staying open. And so when the stomata are open, the water potential drops, and you have a pretty big difference between pre-dawn and midday. And this strategy is exemplified in the literature by junipers. Junipers are known to be drought tolerant and anisohydric. On the other end of the spectrum, we have isohydry, where the stomata are much more conservative, much more sensitive. They close much more readily. And so the difference between pre-dawn and midday there is much shallower. So we've got, just remember, we've got a kind of a deep U on the left and a much shallower U on the right. And that's because of that stomatal control. And isohydry is exemplified by this two needle pinion here. Those are the ones that died. A lot of them died in that 2002-2003 drought. So this difference between pre-dawn and midday tells us about how risky the plant is willing to be in order to acquire carbon. And for this section, I really have the question here of how to disentangle the physical and the biological controls on water potential. Because water potentials are inarguably affected by the physical factors, by the environment. But there's also the imprint of how the plant is choosing to respond to that. How can we disentangle this? And my questions here were, does a creosote's hydraulic regulation vary over time? And what are the environmental drivers associated, or what causes this variation if it's happening? And I have to take a little detour here to talk about how we measure water potential. The classic golden, accepted method is using this pressure chamber. I just toured Ashley's lab. She has 3 of them. And it's a manual measurement, and you need pressure. And how it works is you have to destructively sample the tree, take off a little stem, branch, or leaf, put it in the chamber, and then squeeze it positively with pressure until water comes out of the cut stem. You have to do this. Get up at 3 o'clock. Do this at pre-dawn in the dark. Try not to blow your own eye out because your eye is over this cut end while you're pressurizing it. And it may be some people don't have a lot of trouble with this because their plants are nice, happy, wet plants. These plants that Ashley and I deal with, they go down to -6, -7 megapascals, sometimes -9 for me. So that's a lot of pressure. I'm always worried about running out of gas in the tank and also blowing my eye out. So there's the troubles with that method. It's destructive. You can't do it that often. It's just hard and labor intensive to do. I also had the luck of in being able to borrow some automated stem cyclometers, which at the time were relatively new to the ecosystem. I mean, people knew about them, but no one had really very successfully deployed them. And that I found out quickly is because they're also finicky to use, but in a different way. These ones you can leave on the plant, and they can take measurements for you every half hour if you like, which is great, so much more data. But you have to calibrate, install, maintain, and then once you get the data, you have to then know what to do with that much data and how to clean it, because there's always going to be kind of like erroneous measurements in that time series. That rich time series is what I needed to answer my question of how to disentangle the physical and biological effects on water potential. This, for example, is just my manual measurements. I went out once a month at pre-dawn and took a bunch of measurements. And this is a seasonal time course. You can kind of see that in the spring, water potential was high. It dropped during the pre-monsoon. That's a really hot, dry period before the monsoon comes. Monsoon, there was kind of at least one event where water potential went up again, came back down, and went up in the fall and winter. Great. That tells me something about the environment, right? The climate was different at these different times, so, the plants are kind of like just following the environment. When I add my time series from the stem cyclometers, there's just so much more information in here that can tell me about how the plant is regulating its water potential in addition to the environmental physical factors the plant is unable to escape from. And so here, if you look at the dark green is the pre-dawn and the light green are the midday. Before I showed you those U curves. And in this time series, you can tell that in some places, the dark green and light green are not that far apart from each other. That's that isohydrate. That's that shallow U. And then in other time periods, like in the monsoon of 2016, you can see that drop between pre-dawn and midday to start to increase and become pretty big. And that's that deeper U that marks anisohydrate. So, in addition to just looking at this, there is also a quantitative metric of water potential regulation developed by Martinez, Vialta et al. That I use as well. Again, it's a simple linear relationship where we put midday water potential on the y-axis, pre-dawn water potential on the x-axis, and look at the slope. And because I had these stem psychrometers, I was able to look at that slope at different times of year and not just throw all my data points onto one big graph. And so during the dry season, that pre-monsoon, it looked like the plants were isohydric. The slope was less than 1. And during the monsoon in September, the slope was greater than 1. And so that is indicative of extreme anisohydrate. And there are also times of year, like the fall, where the water potential, where that slope is basically parallel to the 1-to-1 line, and it's also anisohydric. So I was able to do this by cutting my data into these chunks, but I also built a hierarchical Bayesian model to look at how this slope changes over time and what factors might influence how it changes over time. And then I used that model to predict the slope sigma for every single day out there at my site. And I found that water potential regulation varies quite a bit and quite rapidly over time. In this figure, so the higher values are more anisohydric or extreme anisohydric, as shown by the maroon, and the lower values below the 1 line are isohydric, shown in the peach. I want to highlight a couple different seasons for you. This isohydrate, the peach, shows up in the pre-monsoon, and towards the end of the pre-monsoon at that. So they're behaving really conservatively at that point. But this behavior also shows up in the winter. And let me tell you, the winter conditions could not be more different than the conditions in June at that site. Those are very different physical drivers, but the biological response to those drivers is actually the same during these 2 periods of time. And if we look at the other end of the spectrum, the maroon that represents the extreme anisohydrate, the tail end of spring going into the early part of the pre-monsoon is this long chunk of extreme anisohydrate, pretty continuous. And we also see that, but less continuously, in the monsoon, and that's kind of more episodic. How do we explain these patterns? Well, from this data set, I gathered that this extreme anisohydrate that happens in spring and monsoon is a strategy adopted to supply water for photosynthesis and transpiration when growth is possible. So when water is available, the plant is going to do what it needs to do, let its stomata open, let its midday water potentials drop. Because while the going is good, it needs to prioritize that water uptake, because that water uptake means it can photosynthesize. On the other hand, during the isohydrate that we saw in 2 very disparate time periods, the pre-monsoon and the winter, those are both not very conducive to growth. They're either limited by too little water, too hot, or too cold. Creosote are pretty cold sensitive as well. And so this very strong, very conservative regulation of stomata regulation of the water potential is occurring because growth isn't going to happen anyway. So you might as well save your ammunition for another day, preserve that transport capacity, don't let all that air get into your conductive tissues, and save that for when the rains do come and conditions are going to be good again. So that was kind of the last chapter of my dissertation, and I moved on, went to Utah, and I met Steve Cannonberg there. We overlapped at Utah, and he was very gracious enough to really share data and let me work at his site. This is, in case you didn't know, this is what Bears Ears National Monument looks like from the top of his Equivariance Tower out there. And when I first got to campus, the thing that I did was I emailed Steve and said, let's put together a session for next year's ESA. ESA 2020, it's going to be fantastic. It's going to be in Salt Lake City. All kinds of people are going to come. We're going to invite Ashley. We're going to invite Martinez Vialta. We're going to invite all these people. We're going to invite them and give talks, and it's going to be fantastic. And it was fantastic, but it was COVID. So slightly less than what we anticipated. Nevertheless, we got together, had a great session, and then wrote a review paper on this idea of anisohydrate being a trait versus a response. And it all came down to that time scale again. If you're thinking about kind of slow time scales and like pedoclimatic gradients, plant traits that don't change from day to day, like if you measure like leaf area, it's not going to be different substantially tomorrow versus today. That's kind of this trait syndrome approach versus this fast time scale that I was working in, thinking about meteorology, how quickly VPD changes and light changes, and these plant parameters like stomatal conductance and water potential that also have very rapid responses this kind of response-based approach, like how do we merge those together in order to understand better how to and predict how the plants are going to respond to drought. And so, yeah, Ashley and I are co-authors on that paper. And this kind of also, was COVID, that we were home all the time. Writing this paper was really useful. Connecting with this group of people was really useful. That sparked my thinking deeper into what do we mean by anisohydrate. In the last section I talked about anisohydrate. Oh, it's happening during this time and also this time. And then, but junipers are the species that are commonly thought to be anisohydric. They're so drought tolerant, they don't die even though their coexisting pinions are. Yet, during a drought experiment, junipers were also found to have very tight stomatal control in order to avoid xylem cavitation. And so this Anisohydrate framework, which was originally conceived as a drought response, doesn't really make a whole lot of sense because plants are rooted in one place. Again, they can't move. So whatever strategy works during the drought has to also work for that plant during not drought, which hopefully is more of the time, right? So how does this framework apply across all conditions? Are we thinking about this perhaps, could we think about this in a new way? And I was also thinking about how this connected to global carbon fluxes, because in these drylands, it is known that the interannual variation of the terrestrial land sink is actually largely attributable to drylands. And that's not just because drylands are very unproductive when it's dry, but also includes rapid vegetation expansion and productivity in unusually wet years. And when people such as Natasha McBean have taken these vegetation models and matched them to data from dryland tower sites, they found that the models are prone to underestimating, not overestimating, but underestimating carbon uptake in drylands. And what time, particularly during the monsoon at low elevation. So this time component comes into play again. So my research questions for this section include, does the relationship between midday and pre-dawn water potentials vary over time in Utah juniper? Do the drivers of hydraulic regulation or water potential regulation differ from the drivers of gross primary productivity, which is kind of carbon uptake? And lastly, is there a relationship, and if there is, what is it, between this hydraulic regulation and GPP? And to answer this, whereas previously I had creosote shrubs, if they were at an urban, they were inside a city, and we didn't have any eddy covariance, I was able to piggyback onto Steve's eddy covariance site here in southeastern Utah. And so this research was made possible by 3 key ingredients. The first is that we were in an early successional pinyon juniper woodland. That was mostly a juniper woodland. It was 92% of the tree basal area. So a very toy ecosystem, and that toy ecosystem was necessary because I was trying to connect plant level measurements with the stem psychrometers to the whole eddy covariance tower footprint measurements. So you really need a toy ecosystem to do that. The second ingredient is that Steve wrote a grant and got this Ameriflux tower deployed in June 2019. And last year, how time flies, I can't believe it was only last year, I drove up there and we installed 14 stem psychrometers onto 7 mature individuals inside the tower footprint. And Steve maintained them from May until November. So we got a whole growing season. And the last ingredient was actually totally luck. 2021 was a spectacular monsoon year. My house down in Tucson, my backyard was flooded. It's like, what even is, I don't understand what happened, but it was very, very unusual. And so even though this site is at the very edge of the North American monsoon, they got a lot of rain, and that's what I was looking for because I wanted to see the difference of what the junipers were going to do when it was really dry versus when there was water available. And so looking at the water potential time series here at the top, the brown squares this time represent the chamber measurements. Steve didn't always want to wake up at pre-dawn, and I 100% don't blame him. So those brown boxes are supposed to be somewhere between the pre-dawn and midday, and they generally are. And we see that the water potentials were low in the pre-monsoon, and that there's these 3 kind of key pulses that happen during the monsoon, reflecting the 3 kind of major precipitation inputs. And then in the fall, water potential climbed again. And we see a really similar pattern in the GPP on the bottom figure. Okay, so So to answer my first question, just kind of adopting the approach of let's just look at the data, see what it looks like. By plotting the midday versus the pre-dawn in the 3 different seasons, I can just see with my naked eye that there is indeed a shift in that slope, that sigma term, over time. It's pretty parallel in the pre-monsoon and in the fall, but in the monsoon period, there's kind of a parallel to the one-to-one line section, and then there's like a really steep section. Oh, and in this case, the color axis is the soil moisture at 10 centimeters. And again, using the same approach I did before with the hierarchical Bayesian model, I was able to then figure out what sigma, that slope term, was for every single day. And the color scheme is similar here as previous. And I'm able to lay that, this time, I'm able to overlay that with GPP on the second axis. So that's the black line down below. GPP and sigma actually responded to the environmental variables very similarly. They both were highest under a combination of very dry air and very wet soils. So that combination of plenty of water in the soil and a dry air that's kind of sucking the water from the plant, that was when we had the highest sigma, so extreme anisohydrate, very relaxed and open stomata, and also a lot of productivity because the stomata are open, so the CO2 can go in, right? And lastly, for my third question, how are sigma and GPP correlated? Well, looking at the last figure, they're positively correlated. They're positively correlated for the whole season, but that positive correlation is primarily driven by the variability in the monsoon season. So kind of more anisohydric, the stomata are more relaxed and more open. This makes perfect sense from a theoretical point of view, but we were able to show this empirically that is positively correlated with greater carbon uptake for the whole ecosystem. Now, if any of you were examining that first time series really closely, here it is again. You might notice that the peaks of these don't line up very well. So the peaks of where the extreme anisohydrate occurs and the timing of when GPP peaks don't line up perfectly. And since I'm very interested in the timing of things, I was like, what is going on? Why don't they match? Shouldn't they match perfectly? And I actually took an offset, so I offset those 2 time series and found that the highest correlation coefficient actually occurred with a 1 to 2 day offset. So GPP actually peaks first, and then a peak in sigma happens after 1 to 2 days later. What is that about? My hypothesis here is that extreme anisohydrate, that plant response, allowing for more relaxed control of the stomata, serves to prolong elevated GPP. So that GPP hits the peak first, but to keep it up there, because otherwise the soil moisture traces drop pretty rapidly, requires the midday water potentials to drop a lot in order to keep moving that water up the plant. And so this extreme anisohydrate prolongs elevated GPP and efficiently extracts soil moisture after these large rain events. And that caused me to kind of have a shift in my thinking here from anisohydrate being kind of a state, whereas in my previous vignette I was talking about, during this time of year it was anisohydric, during that time of year it was isohydric, to thinking about anisohydrate overall as a strategy. And in particular, it's a flexible, year-round strategy, as befits plants that can't move, right? They might experience highly variable environmental signals, but they can't move. And it's found in seasonal dry ecosystems. Again, very pronounced when there's like a dry period and a wet period. You can see this change in strategy, most evident. And that this is a strategy overall adopted by plants in order to enhance productivity whenever the conditions are suitable. Right? It only makes sense to have the relaxed stomata in order to take up a lot of CO2 as a substrate for photosynthesis if the other conditions are suitable as well. That strategy doesn't make sense when it's really hot and dry or it's really cold and photosynthesis shuts down anyways. All right, on to my final slide here. My final concluding message I'd like to leave you with today is that rather than being although they are rooted in place, rather than being kind of at the mercy of Mother Nature, plants do have biological control over how they respond to the environment. And this is particularly evident at those intermediate or synoptic timescales. Understanding how and when plants exert this control is critical to predicting the magnitude and also the timing of dryland carbon and water fluxes.